What if I told you that curing all genetic diseases could be as simple as replacing an item on a shelf? Well, let me introduce you to CRISPR.
First, let's identify a few terms. Clustered regularly interspaced short palindromic repeats, or CRISPR, are repetitive sequences of DNA. CRISPR-associated proteins, or Cas proteins, chop up each sequence of DNA. Usually in CRISPR application, the Cas9 protein is used. Now, let's talk about why we care about these DNA segments and the protein. Let's go back to 1987, when the useful tool was originally discovered through an E. coli bacteria fighting off a viral infection. When a virus invaded the bacteria, a Cas protein cut out a portion of the virus's DNA in order to keep it within the CRISPRs. This essentially allows the bacteria to keep a picture of the virus within its memory so it will not be welcomed again. Because that strand of DNA cannot move within the bacteria, a copy of RNA is created. This RNA will move around the bacteria along with the Cas protein, acting like a cop on the manhunt for a criminal. If a strand of the virus is spotted, the Cas9 will react swiftly and catch the criminal, which in this case means eliminating the virus.
How does a bacteria's defense against evil viruses help us? Well, we can take this genome editing tool and apply it to anything we want. Here's how.
A scientist would need to create a sequence of RNA that matches the specific gene they want to alter. This RNA is referred to as the guide RNA, as it will guide the Cas9 protein to the gene. Upon arriving to the target DNA, the Cas9 will cut it off. Naturally, the cell will try to join the two ends of the DNA together from where it was cut, but we don't want this to happen as bases often go missing. To prevent this, the scientist should add a template of DNA onto the Cas9 so the cell will add it to the gap.
You may be thinking to yourself, is this too good to be true? Well, recent results from a CRISPR experiment show that it isn't.
42 out of 44 people suffering from transfusion-dependent beta thalassemia and 31 out of 31 sickle cell disease patients were completely cured of their disease through use of CRISPR technology. 3 years later, there has been no recurrence of the diseases or symptoms.
Due to its high rate of success in organic technology, CRISPR might be the future in eliminating genetic diseases such as sickle cell disease, muscular dystrophy, and cystic fibrosis. It may even be able to vanquish the most horrible disease of all, cancer.
The reason cancer spreads so quickly is due to a mutation in DNA that causes undivided cell growth, which CRISPR may be able to cut out. CRISPR even has other applications, such as growing bigger fruit.
Though CRISPR is very promising, we need to remember, as with all other scientific discoveries, that it needs to be implemented safely and ethically.